Last chapter, we talked about the cell, and certainly we talked about the cell membrane, the plasma membrane. This chapter, which is fairly brief, but it talks about the cell membrane itself and the key components of it, and how those key components allow the cell to be what we keep on saying as selectively permeable. It allows some things in, your cells allow some things in and not others, and also allows a adaptability with regard to the cell, and this is split-second adaptability. That's what we mean, I guess, or a portion of what we mean with the fluid mosaic of the cell membrane. I kept saying in the last chapter that the cell membrane walls off the cell from the outside world, so it defines the cell as inside and outside. But it isn't quite that simple because the membrane is constantly moving. What you want to, we talk about this mosaic model, but what you want to think of is picture yourself on the second story looking down at a whole mass of people. Those whole mass of people would be representing your phospholipid layer. It's a solid phospholipid layer, so it's a solid group of people who are constantly moving around, who are constantly jockeying and moving and maybe talking to this person there and that person there. That's what happens with the cell membrane. You have a solid mass of that biphospholipid molecule, but it is constantly moving and rearranging itself and allowing for things to be added to the membrane and also allowing for the membrane to repair. So we do have this concept of a fluid environment, fluid mosaic, which is, you think of the word fluid, but you also think of it as solid. And this is what allows the cell to change its shape, but keep that border, and also allows for things to be embedded in it, like proteins, to be embedded in it and make sure those proteins are in the right spot for where they're needed at that time. What you have here are various images. I couldn't find exactly what you had in your book, but I have something that is very similar. In the upper left, you have your phospholipid molecule, where you have your portion that actually doesn't like water, and then your portion that likes water, which is the lipid, the red portion, and your portion that doesn't like water, the hydrophobic, which is the long carbon chain with hydrogens. And it is in two sections, and then what you're seeing toward the right of that same picture on the left. So on the left, the upper left, you have two parts to it. You have the water on the outside and then the membrane. And that is a pretty good representation of the membrane in the fact that it kind of looks just like a bundle of thread, but the thread is much more organized than you might think. And then in the bottom left, you have what's known as an aquaporin, and these can be entered into. This is something that can be at need, as need be for homeostasis, can be injected into, can be inserted, not injected, inserted into the membrane to allow water to move in and out. It's a water channel. And then on the right, you have various different proteins that are embedded, whether they are peripheral proteins or integral proteins. All of these happen to be integral because they are from the both sides of the water, the external fluid and the internal fluid. But there can be proteins that are just on the periphery also. And we'll talk a little bit more about them. This is information about, just kind of refreshing your memory maybe. Plasma membrane is the boundary that separates outside and inside of the cell, and this is, we're talking about animal cells. In plant cells, you have your cell wall. Plasma membrane exhibits that selective permeability And what that means is some things can enter the cell, some things not. They generally have, they enter via a channel, or they diffuse in, or there may be reasons why they may be lipid soluble or something like that. They may be soluble through the membrane. The membrane is made up of phospholipids, and phospholipids are this word amphipathic. Amphipathic. This is the word that means that a molecule is hydrophobic and hydrophilic and hydrophobic. Hydrophilic, the portion that actually attracts water. Hydrophobic repels water. And that causes the arrangement of the molecule, so that the molecule arranges in that biphospholipid arrangement. And then you have this word mosaic, which is the fact that it is made up of multiple units of the same thing, certainly creates a whole, but it does have this dynamic capacity to it. It's always the plasma membrane, but it never is completely done being itself. It's difficult to describe. I kind of like my scenario of looking down at a group of people and the fact that they're all continually people, they're all continually there, but they're actually moving around quite a bit. And then you have your image of your molecule to the far right. Couple of the things that the membrane can do, you can have lateral movement of phospholipids, and one might want to have lateral movement if in fact there's a need, if there's actually something that is near that phospholipid. So we have these, remember we're going to have these proteins embedded. And if there's a reason that you need to move that protein closer to where it may be able to perform its function, there's this lateral movement. Flip-flops can happen. It's actually kind of a good thing that you have this movement going on. So you have lateral movement, which is very quick. Flip-flops about once a month. You can have some fluid movement where you have saturated and unsaturated fatty acids. Fluid movement, you find these unsaturated membranes in fish. It becomes a very, not because they're in the water, but it becomes that helps them be very, helps them adapt to their environment. I guess it is because they're in the water when you think about it. And then you have very viscous, if you have these saturated, viscous meaning thicker. Cholesterol tends to stabilize the plasma membrane. It's one of the reasons why we have cholesterol embedded in, is otherwise the membrane would be a little bit too fluid, so the cholesterol is in there to form a little bit of stability, depending on whether the cell needs that stability. So all of this is an adaptation of that individual cell to the environment it's living in. Proteins and their function, we talked about this a little bit in the beginning, and here's the same picture again, and then I have it listed on the next slide. So you have some of the selectivity of the membrane is due to specific proteins, to specific transport proteins. And they are embedded in the membrane. When we say embedded, that means they have a relationship to the outside fluid and the inside fluid. Proteins also tend to have a hydrophilic, hydrophobic portion to them, and so they tend to be embedded in an arrangement where they will have the opportunity for the water to come close to them, or for them to repel the fluids. They can be either integral or they can be peripheral. So proteins can span, and I'm looking at the bottom portion of the dialogue here, is you can have peripheral proteins that are on the outside on that extracellular matrix, embedded in that, or they can be all the way through the membrane. And they are also, integrals are also called transmembrane proteins. You have various types here. You have a transporter protein, which we think of that as being a channel to allow a lot of times ions to pass through. You have a protein that is designed for one particular enzyme. When that enzyme is there and acts as a catalyst, it may change the shape of the protein, and in changing the shape, it allows for something to actually happen on the outside of the membrane. You have cell surface receptors. What these are embedded in the membrane to identify that self as self, or to be able to take in a particular product that the cell needs. It's embedded in the membrane with a receptor site on the outside, so on the external portion of the cell, to allow something to actually make contact with the cell and be taken in if need be. You have surface identity markers. These are better to think of as how your cells know that they're your cells, how you identify self, and also how your cells know that they're a blood cell as opposed to an epithelial cell. Some proteins are embedded for adhesion, and some proteins are embedded in the membrane to attach to the cytoskeleton. Remember, the cytoskeleton is inside the cell. And then this particular slide is just to review the types of proteins. It might help you it might help you if every once in a while you pause the lectures and make some special notes. So transport proteins, you can think of those as, remember, those are the channels where you have ions passing through, so transport proteins. Enzyme activity, where you have, and you have another image here, you have a specific enzyme that when it comes in contact with a protein, it can actually cause something different to happen on the outside. Cell-to-cell recognition, generally a glycoprotein. Intercellular joining, so allowing cells to join. And then at the bottom right, you have a protein that is in the cell membrane, so the plasma membrane, but actually allows that external matrix, that collagen fiber, or the fibers that are part of the external matrix complex, to attach to the cell. So the cell actually attaches to its own environment. One might think that extracellular matrix, the ECM. So at least 6 different types of proteins: integral proteins, transmembrane proteins, those proteins that are embedded in the plasma membrane, for you to kind of get an idea of which is which. Then the role of carbohydrates, and carbohydrates are many times attached to proteins. They bond well. Surface carbohydrates are often found on the surface of the plasma membrane. What they do is they can cause cell-to-cell recognition, and so the carbohydrates covalently bond to lipids, and they are often used to identify types of cells, so that particular carbohydrate arrangements. Remember, carbohydrates can be very long, and in order to understand what type of cell we have here, that the carbohydrates are embedded to actually give that cell its own identity. And this is often used by viruses. Viruses can recognize a good host cell by the carbohydrates that are on the surface of it, and bond with those carbohydrates and then invade the cell. And that's the image you have here. The example is HIV and the receptor that it tends to bond with, the CD4. And so if that can be changed, it can infect. And we'll move on.